go ahead and put, we'll use this. Brandon, you're going to need one of these. All right. Okay, so we know it's deoxyribonucleic acid. Let's put inherited genetic material in the form of 2 strands of nucleotides. Okay, so lots of stuff in that one sentence. So we know the identity, kind of where it comes from will be more explicit in a second, but inherited genetic material, it's like that whole genetic material thing is what they'll write in lieu of DNA, just so it's inferred that it's passed on from your folks or from a folk to their clone offspring. So in the form of 2 strands of nucleotides. So here are the monomers. And DNA is a polymer of nucleic acid. So that's the organic molecule. Alrighty, so where did you get yours from? So what if you're asexual, a product of asexual reproduction?
You're a clone of your parent.
Right, okay, so where's yours? Okay, so in organisms, it's passed on from to its clone offspring. So they may ask you a question, how would the arrangement of bases of an offspring compare to its parent if they're a product of asexual reproduction? So you would say it's the exact same, right? So passed on from a parent, asexual, to its clone offspring, or to half of their genetic material their offspring. All right, so it doesn't mean to say, okay, I think I'll just give you this half, so we know it's a little bit more orderly than that. And we'll talk more about sex cells and how that works and all that other business in the next unit. But so we know how you get it's inherited. Not acquired. Okay, so what do I mean by that? Right. So if you decide, and this is important because we're going to talk about DNA and its role in evolution and change. Evolution is such a scary term for some people. It's like, ooh, evolution. No, evolution all simply means is the species changes over time. Their genetic material, it might be that one individual has better genes that help them to be more suitable in an environment, and maybe another offspring doesn't have that. Maybe they were born with an albino gene and they stick out like a sore thumb, and a predator comes and chomps them down, and takes them out of the gene pool. So some of the genes are good that you're going to inherit. Some of them might not be so good. But either way, they have to be passed on. Otherwise, if you do anything, like let's say you get really buff and you're really getting very muscular, and you can't pass on those big, bad, muscular, buff muscles onto your baby. You have this little Popeye baby that's born. Doesn't work that way. So anyway, it is important to say that they're inherited and it's not acquired. Okay, so next. Where is it located? Where? And cells. Okay, so first of all, we know that it's one of those things that all cells have to have. So we know that no matter if you're prokaryotic or eukaryotic. So if you're prokaryotic, like all the bacteria, where would you find the DNA? And cytosol, cytoplasm, just kind of out floating around there. So cytosol, cytoplasm. Okay, so where would you find it for all the eukaryotes? And the nucleus, right. And then we just found out that plus 0.5% in, or 1.5%, was it?
Yeah, it's 1.5.
Yeah. Approximately 1.5, it depends on the species, I believe, but in mitochondria. And to be very honest, we can include in there chloroplast too, for some. And we know we don't have chloroplast, and that's why they didn't mention it. But for plants, chloroplasts also have a little DNA in them. So that's evidence that they might have been living things at one point. They can reproduce on their own. They have their own ribosomes. They have their own DNA. It looks like that most likely they used to be bacteria at one point. Okay, so that's what they are, where they are, what's its function. So what's the whole big deal? So what's the function of DNA? Must be pretty important because you inherit it from your folks, and you wouldn't be here if you didn't have it. Is it what? Yeah, what do you think? What's the big deal?
It helps with our structure.
So like it determines who you are and like how you live.
Okay, so I hear there's all the rumblings of you need it to live. It's going to determine some traits, give you some diversity. It's going to determine maybe what you're able to do, what traits you might have. All of that basically is protein, plain and simple. It is all protein. When you look in the mirror, protein. I mean, it just is. So the function is DNA is divided into sections called genes. And each gene determines a trait, which is a protein. Now, it didn't mean that it would translate just into like the size of your foot or the color of your eyes. I mean, that's true, but it might be, there's tons of enzymes that actually are involved in all these chemical reactions that determine those things. So if you're missing a certain enzyme, it might actually cause you to have a certain genetic disease. There's one mutation in the color gene that gives you albinism. So there's like two genes. You can let's say you have the darkest parents in the world or the lightest parents in the world, but you also got an albino gene. Now you are super light if you got light parents. And if you had really dark genes, now you're albino, but you might not look as super light person over here that got the really light genes. So when you look at people and albinism, it's funny. It's like if you get the albino gene, which was a mutation in the code, then it screws around with your protein, like what you're going to look like. So I don't know. We're going to see it. There's some really cool things that can happen as far as that goes. But yeah.
What about the people who like
They have extra melanin.
They have what?
Excessive melanin.
Excessive melanin.
I should say the opposite.
Yeah. It's just a gene. It's just, and I don't know if it's dominant. Now, I will say it's darkness is always dominant over lightness in people. That's true. So if you're lighter, we'll find out with genetics. That's where it gets a lot of. We won't get too much into it now, but it gets a lot of fun. You can look into. We find out there's lots of genes that determine, like, for example, skin color. There's like 3. So you may get a dark version here. You might get light. So then there's like a whole like bell curve of color of where you can fall. It's pretty neat.
A little bit of pink. Also, almost like whatever your design is, you take off.
Doesn't matter. It's not sex linked, and that's why I'm saying it is not a sex linked trait. It has nothing to do with your gender. Yeah. So, all right. Yeah, Brandon, go ahead.
I've always been wondering my whole life why. So both of my parents are fully Hispanics, but I came up to look like Canadian. I don't know why.
It has to do with. It would have to do with the eye shape. When we talk about genetics, you'll see there's different versions of eye shape, and you'll see if they're recessive or dominant. And right off, I can't remember what's recessive and what's dominant. I would say that it's probably recessive because when you look at your parents, if they don't have the same eye shape as you, they probably were both carrying a recessive gene and you just happen to get those. So somewhere down the line, somebody looked just like you. Okay, so structure real quick. So we know there's 2 strands of nucleotides twisted and bonded together. And we call that 2 strands twisted a double helix. Yeah, or a twisted ladder model of DNA. All right, so I'm going to take one color. We're just going to draw it out. It doesn't really, actually, I'm not going to use a color for this. We're going to do phosphate first. I'm going to put a P. I'll just label it one time. Now, even though I'm making mine look like the little Sunhouse Garage nucleotides, for this DNA model right here, there's no circles. But it's going to be something here on the outside. I'm not going to tell you what it is yet, just if you can figure it out. But it's going to be out here on the outside. Okay, so it's always bonded to a sugar. So this is where I am with the color. And it's a 5-carbon sugar called deoxyribose. So that's why it looks like a pentagon. Okay, 5-carbon sugar. Deoxyribose. Okay, so that's going to be the second thing that's on the outside of that model. And then in the middle, so sugar phosphate is going to be one of these, either the black little star beads or the band. Okay. And then we're going to put these nitrogen bases in the middle. So grab another color, and we're going to put it right here. And I'm going to put an A, and I'm going to make it adenine. Okay, yep. So what is adenine? Ivan said it earlier. What is adenine's partner going to always be? Thymine, right. And it's going to be bonded together by hydrogen bonds. Now, hydrogen bonds, when you take chemistry, Miss Wall is going to tell you, well, it's not really a true bond. So hydrogen bonds are more like magnets. So when you put a positive end of one magnet to a negative end of another magnet, what happens? But you can feel the force, right? There's a pull that pulls them together. That's what hydrogen bonds are. So it's like a pull when they're next to each other. They want to combine like that. And then they have to be pulled apart. So with an enzyme. So we're going to put the thymine across from it. So let's do that. T. And there's the sugar. And then we'll do the phosphate. I'm hitting the black and it's not turning black. There we go. Okay.
What do you
Are
do?
hydrogen bonds and phosphates supposed to be the same color?
It doesn't matter.
Okay, I just.
Yeah, it really doesn't. So for your models, you have to have the same color for the phosphate, the same color for your sugar, and then you have to have the same colors for your A's, your T's, your C's, and your G's. Okay. So now let's see if you can figure it out. What part of the model is the phosphate and what part is the sugar?
The sugar is the red.
Okay, so I heard sugar is the red and the phosphate is the black. All right, how many people agree with that? Sugar is here, phosphate is here. Okay, and look at the model.
Oh. I see it.
Okay, how many people say that the sugar is on the end connected to the nitrogen base, phosphate's in between?
That's what I'm saying.
Yeah.
How many people are like, I have no idea? Okay, so let's draw the next pair and then let's see if you agree or disagree. Okay? Oops, go back. Where are you? Where are you? Oh no, where'd you go? Where'd you go?
That's it, right?
Where is it?
No,
I mean-
it hasn't been read.
That's mine for the other class. I just started like drawing on a page. It was not very smart on my part. Dang it. Oh, there it is. Okay, so here we go. Okay, so let's do G guanine.
I'm kidding.
Cytosine. Can y'all read that yellow or do I need to go over it?
Cytosine, yeah.
Can you see that?
Bless you.
What is the word?
This one, Guanine. Okay, so now after you get those, how many pairs of nucleotides do you see? Pairs. 2 pairs, but you have 4 individual nucleotides, right? So one of these is a nucleotide. Okay, because it's composed of a sugar, a phosphate, and a nitrogen base. So Francis is right about the 4. Your model is asking you for 6 pairs. Okay, so 2, 4, 6, 8, 10, 12. So it only has to be this big. The only reason that they wanted to make it bigger is they wanted to make it look nicer, they said, plus they wanted to try to get a full twist in there. So you have to show yours twisted. You also, it has to be freestanding or you have to be able to hang it up. And it has to be neat. You have to show the bonds in between. So how many hydrogen bonds are always between G and C? 3. And there's 2 there between A and T. Y'all have to show that. So you can do it with a marker, you can do it with toothpicks, you can do it with wire, whatever you decide. So as you're planning it, think about what could I connect these together with in the middle. So now let's go back to the question. What's the phosphate and what's the sugar? How many folks say that the red band is the sugar and the black is the phosphate? That was the first group. Okay, how many people say that the phosphate is this in between and the sugars are on the end?
Actually, I think the phosphates are on the end.
No.
I think the phosphates are on the end.
You can change your answer.
Oh, Jesus.
Okay, so what do you notice? What did we draw at the ends of the nitrogen bases up here?
Phosphates.
No, we didn't.
Oh, wait.
Sugars.
Oh, there. Oh.
So the sugars are the black beans.
I always thought it was the red connects to everything else in the sugar.
So the phosphate is alone by itself right here. And it's a spacer in between. So the model in the very back, that one's confusing. Usually it's great, but it looks really fancy. But usually that one confuses people because I draw the phosphate as a circle and they say, oh yeah, the phosphate's the circle, and it's not in that model. So one reason you're going to have a key is because we can look at your key and say, okay, that's what these parts are. So their key is correct, but usually when people see it, they get it confused. Okay, so you may have circles or you can use a band. It doesn't matter. These have to be the same candy or same material, whatever it is you use. These have to be the same, and then the phosphate has to be the same. So if you have a blue ribbon and a black ribbon, you can't do that. You have to have the same kind of ribbon. It's in color. So think about that as you plan, because you're going to have time in a minute.
What does that do?
We're going to do it tomorrow. You have 45 minutes in class, so you have to have kind of a plan. And then I'll have some stuff, but if there's anything that you want to supplement with, because you can hang it up or you can stand. It doesn't have to stand necessarily.
They did that in class?
I did it in 45 minutes.
Wow.
Yeah. Yep. So that was, who are these people? Jaleesa and Elizabeth. So, it fits in there.
She broke it.
It fits right in there. The little straws. Okay. So that's it as far as that part goes. So we just need to label the nitrogen bases. So let's just do that. So we say that, go back. This and this are nitrogen bases.
I don't have room to do that.
And here's the honors curriculum part, is that A and G are called purine bases. And C and T are pyrimidine bases. So ATP is adenosine triphosphate. It had an adenine, it had a ribose, and it has phosphate. So it is actually it had purine bases. It is actually a nucleotide. And there's one called GTP too, and it has guanine in it instead of adenine. Okay, all right, so what time is left? You've got several things you can do. One thing is you can get with a partner. If you have a partner in here that you want to build a model with, you can do it by yourself or with one person. You can talk about that. The rubric is the back of the first sheet. Okay, so check that out. And you can work on that. You can also work on your fact sheet, and you can also work on your video. So it's all up to you. And I'll be here until 4:15 today.
We do